Welcome to Gramene webinar. Today, I would like to talk to you about various genomic resources, data, and tools we have available for tomato researchers at Gramene. So this is Gramene homepage. We have our resources available under two portals. So first is the genome portal. If you click here, the genome page opens up and you can see we have about 39 plant genome browsers and plant genomes available at Grameen. You can choose your favorite plant species tomato here, and then you can land into tomato genome page. In this page, we have organized various resources available for tomato. Here you can see example region of genome browser and then go to desired region. You can see karyotype view. You can also see example gene trees and generate your trees here. So you can do comparative genomics. We have annotated genes and their detailed information, sequences, cDNAs, and so on. We also have information here for variation data. For tomato especially, we have about 10 million SNPs and other variants from 84 tomato accessions. So data source for this is 100 Tomato Genome Project. You can explore these variation data for tomato. So if you click here, you can land on this page that tells you about the source, and you can see all predicted consequences of the variants. If you like to browse in detail within the context of Genome Browser, you can click here. You can also selectively see consequences of those genomic variations on genes and regulatory regions. Or you could explore genotypes, and here you can explore 84 tomato genomes and how they are uh, affected by those nucleotide polymorphism. In Grameen page, we have various video tutorials, recorded video tutorials that are available that show in detail various functionalities and how you can explore data tools that are under genome portals. Today, I'm going to focus on pathways. So you can go to our pathway portal directly from Grameen page. It can take you to Plant Reactome homepage, or you can come to Plant Reactome homepage directly from plantreactome.grameen.org. In summary, Plant Reactome is a resource for comparative pathway analysis. Currently, we use rice as a reference species. We curate metabolic, regulatory, signaling pathways, developmental pathways now in rice, and based on homology projections, we project pathways for about 62 plant species. We also provide tools for omic data analysis and visualization, and also comparison of pathways between reference and projected species. So I'll explore all these features with you in a step-by-step manner. Again, we have previous recorded tutorials how to use plant reactome for various species. So feel free to go there. Typically, we update our content 3 to 5 times per year. And so if you click on content, you can find release summary. So we have currently release 50, and you can find all the updates that we have made in release 50. You can also find a table of all the genes, all the plant species for which we have pathway projections available. Again, Oriza here is a reference. Particularly for Solanaceae, we have tomato and we have potato pathways available. We are in a process of adding more species, and we are also in process of adding more curated pathways. So please check the updates regularly. Now I explore with you how to browse pathway and how plant reactome pathways are organized. So you can click on Browse Pathways here. That will take you to Pathway Browser. So typically this left-hand side window shows you organization of pathways using enzyme function and ontology schema. On the top right side, you have pathway diagram. You can zoom or zoom out or zoom in into this pathway diagram. Here, we use eukaryotic cell model to depict a pathway. For example, this is a gismonic acid signaling pathway, and it occurs in two compartments, cytosol and nucleoplasm. Again, you see these various color boxes. So we have ways to depict small molecules, we have ways to depict proteins or protein complexes. Also, here is the different type of reactions that we can show within our pathway diagrams. So we can show traditional metabolic reactions, transport reactions, dimerization reactions, inhibition, repression, and so on. So on this side, we have the key for exploring and understanding this particular diagram. On the bottom side, we have various data that are associated with molecules like compounds or proteins, about the expression data and all related data that you can get into. So here typically you have this Oryza sativa as our reference species by default, but using this pull down menu you can select any other species. You can also change the view, so you can hide one or more of this panel and play around with the view. Again, you can look into the molecules, and also you can look into other tabs here like summary. Here is example of expression data. So for reference species rice, we do have EBI Atlas baseline expression data available for Oryza sativa. We do not have this data available for tomato. We would like to work with tomato community if they like to share their data, and we are happy to project it. Again, so now this is a view of tomato pathways. So as I said before, you can select your favorite species here, and you can see that we are able to model based on homology projection the pathway for tomato. Again, in the molecule section, if you click on that, then you can explore various molecules that are associated with this pathway and go into the detailed page on Chabi of individual molecule. You can do similar things for the proteins. So if you click on this protein coding gene, it can take you to Gramene gene page, and from there you can go to genome page. You can see this gene within the context of genome browser, transcript data, variation data, and so on associated with this particular gene. There is another way to explore our pathways within this schema. So rather than choosing an individual small pathway, you can choose a top category. And here you can see all related pathways. For example, all pathways involved in amino acid metabolism. Here is example of carbohydrate metabolism related pathways. And you can see all the list of the pathways that we have so far curated for rice and projected for other species. And you can see where they take place. So as you see, many of those are in cytosol, some are in plastid, other are in Golgi, and so on. Now, sometimes you don't have time to browse. You just want to quickly get into a gene that you are interested in and see if we have anything available in plant reactome for that gene. So here is a case study. So I'm going to use example of COI gene that is required for maternal control of seed maturation. You can go to plant reactome, and here we have a search box. So you can type COI, and this is what you get as a result. So you get the information that we have in plant reactome. So you get that COI1 basically forms a complex with gismonic acid. And then it says which reaction it is, and you can explore further, and you can get into the pathway schema here, and then finally into a specific pathway. So you can click here, and again you can go to pathway browser showing this particular pathway. So this is the COI gene here in tomato, and it shows where it is taking place, if this reaction is taking place in cytosol, and so on. So this is a quick search way. You can also search for compounds. You can search for small molecules, and please explore it. If you have any problems, let us know. Now I'm going to talk to you a little bit about the tools we have for analysis of data. So you can go to our page here. You can click on Analyze Data, and this window opens up here. This is an icon. So if you are in any window and you see this icon and you want to quickly go to analyze data more, you can click on that and it will again open the same window here. So here first I will show you comparison of a pathway between reference species and a project species. So what you can do here, you can say compare Oryza sativa with, and then select a species here. Here you have list and you can choose your favorite species, here in case tomato, and then hit compare. Now you can see this pathway. So basically this pathway diagram is from rice, and what you see here is projection of tomato genes and rice genes on this pathway. So here is the key. What you have here, you have the blue boxes, sometimes here and here. So these blue boxes means that we do have genes in Oryza sativa. However, we are not able to identify corresponding tomato genes. So for dicot species, our cutoff value is 30%. For monocot, we have 40%. So we might have missed using the algorithm we use for identifying orthologs. Now, we have also boxes here that are completely yellow. It means that we found match of rice genes and tomato genes, and number of the genes in rice and number of the genes in tomato are similar. You see also boxes that are half yellow and half blue, and it means that we do find one or more ortholog in tomato. However, number of genes mapped to this particular enzyme in rice and tomato, they are different. So their number is different. So in the bottom panel here, you can explore, you can just explore the pathway. Here is a list of pathways and their matching and so on. What you could do, you could download these results and explore either in Excel format, or if you like to, you can also explore one pathway after another pathway and use the visualization here offered in Calculator. Now I'll quickly go to data analysis. So you can use transcriptome data, you can use also metabolome data and see their visualization on pathway. So again, here you say project. Don't forget to click on here project to Oryza sativa, and then you can either upload your data file or you can paste your data right here. Currently, our this data analysis tool is working very good in Chrome, and you can also use Safari, but other browsers, other web browsers are not so good, and we hope that we will improve on that aspect in future. So here is an example of a public data set from tomato that was generated by Tomato Genome Consortium, and it was published in Nature in 2012. Here is a data source. So I can just paste this data here. It is a tissue-specific expression of tomato genes in various tissues like leaf, flower, root, fruit, and so on. So you can put this data here and then you can see analyze. And again, what you can do, you can choose a top category and see all the pathways related to carbohydrate or amino acid or secondary metabolites and so on, and see what is happening to those pathways. So typically here, if you have yellow color, then genes associated with those pathways are upregulated. If you have blue color, it means that genes associated with some of these pathways are downregulated. Here again, in the bottom, you can have a list of the pathways and the genes, and you can download those results, and you can explore using individual pathways. And here in the bottom, if you have multiple sample data, you can go backward and forward from one sample to another sample, and also have the comparative view. So if you click on any of the pathway you think is of interest here, then you can also see differential gene expression mapped to a single pathway. So this is example of Koschmidt biosynthesis pathway. You see here expression of the genes that are associated with various steps of this pathway. And then if you click on a particular enzyme here, you can open up this window, and here you see the trice genes that are mapped to that particular enzyme, and then corresponding orthologs for tomato that are mapped here. As you see that although there are these homologs, for example, 4 of them are mapped to the same enzyme, but their expression is not similar. And this is often the case with plants and also other eukaryotes that harbor multiple gene family members. And especially identifying candidate genes in plants is very difficult because plants have a lot of gene duplications and large gene families and so on. So we hope that with this particular analysis, our users can shortlist their candidate genes. So this is what we have so far in plant reactome side in terms of the tools for expression data. Similar analysis people can do also using metabolomics data. So you can upload your metabolomics data and you can see those metabolome profiles within the context of pathways. And again, you can download results right here in Excel format. So this was all we have at Plant Reactome. I would like to thank various colleagues from Gramene and our collaborators, which are in various other laboratories. I thank you them very much for their support, and I thank you.